Good day everyone. Today we'll be discussing chromosome abnormalities. So chromosomes, they are consist of DNAs and proteins and is duplicated and transmitted via the reproduction, mitosis or meiosis. So what are the parts of the chromosome? We have the heterochromatin. Heterochromatin stains dark, while the euchromatin stains light. So what are the essential parts? We have the telomeres, they are the chromosome tips. Then we have the origin of the replication sites. Of the centromere, the largest constriction of a chromosome. So that's the part we have here, the telomere and the centromere. So extra our missing chromosomes are easily detected by counting a number other than 46. So identifying chromosome arrangements, such as inverted sequence or an exchange of parts between two chromosomes, requires a way to distinguish among the chromosomes. So a combination of stains and DNA probes are used. So obtaining cells for chromosome study. So any cell other than a mature RBC can be used to examine chromosomes. So for adults, WBCs separated from blood samples or skin-like cells inside the cheeks are usually used. So person might require such tests if there is a family history of chromosomal abnormality or to identify blood-bone cancers, those chromosomes inside the bone marrow. So the chromosome tests are commonly performed from the cells from the fetuses. So the couples who have been diagnosed with a chromosome abnormality can arrange for treatment of the newborn. So the process of is amniocentesis. So the first fetal karyotype was constructed in 1966. So, ano ginagawa dyan? The doctors remove a small sample of fetal cells and fluids from the uterus. So, cells are cultured for a week to 10 days, and typically 20 cells are karyotype. So, the sample, anong sample dun? Amniotic fluid. Before we discuss it's a print 2 about the amniotic fluid. So it is examined for deficient, excess, or abnormal biochemicals that could indicate an inborn error of metabolism. Then ultrasound is used to follow needle's movement and to visualize the fetal. So yan yun, the amniocentesis, they are using syringe to get the amniotic fluid surrounding the baby. And they use ultrasound scanner as their guide. So it can detect approximately 800 of more than 5,000 known chromosomal and biochemical problems. So most common chromosomal abnormality detected is one extra chromosome called trisomy. So it is performed, tandaan nyo to, 14 to 16 weeks of gestation. So most common candidate is a pregnant woman over age 35 because this is the advanced maternal stage and mas mataas yung risk or increase ng risk of extra or missing chromosome on the fetus. So, another reason to do amniocentesis ay to reveal the levels of the fetal liver protein called the alpha fetoprotein and HCG or the human chorionic gonadotropin. So chorionic sampling cells are obtained from the chorionic villi during the 10 to 12 week of pregnancy. So, pwedeng makita dito ay yung mga transverse limb defects. So, yan naman yun. Ginagamit the chorionic villi. CVS does not sample amniotic fluid, so the tests for inborn error of metabolism are not possible. Tapos, earlier results, but greater risk of spontaneous abortion. Tingnan nyo naman, sobrang liit lang nung fetus pa. May ipapasok sa kanya, delikado. and it can cause nga spontaneous abortion. It is slightly more invasive and dangerous. Tapos yan, difference between amniocentesis versus CVS. Amniocentesis, gestation is 14 to 16. Tapos it uses needle. CVS, 10 to 12 gestation time. Tapos ang route niya ay sa vagina. So fetal cell sorting naman a newer technique that separates fetal cells from the woman's bloodstream. It is safer than CVS. So technology timeline in 1923, human chromosome number thought to be 48. Then 1951, detangle chromosomes. 1953, developed the squash and stain technique. 1956, determined chromosome number to be 46. 1956, identified 23 chromosomes in human gametes. 1959, first chromosome abnormalities identified. 1960, phytohemagglutinin added to the chromosome. 1970s, several chromosome stains improved the resolution of karyotypes. 1970s, fluorescence in situ hybridization or the FISH was developed. 1990s, spectral karyotyping combines FISH probes to distinguish its chromosomes. So chromosomal shorthand indicates the chromosome number, sex chromosome, constitution, and the nature of the specific chromosomal abnormality. So 46XY, that's for normal male. 46XX, that's for normal female. 45X, that's the Turner syndrome, it's for female. And then we have 47XXY, we have the Klinefelter syndrome for male. We have 46XY, then deleted yung 7Q, a male missing part of the long chromosome arm of chromosome 7. Then 47XX plus 21, a female with a trisomy 21 Down syndrome. 46XY, translocation 79, a male with translocation between the short arm of chromosome 7 at band 21.1 and the long arm of the chromosome 9 at band 34.1. Then we have 48XXYY, a male with an extra X and an extra Y. Then 47 triple X or the triple X syndrome for female. We have 47XY plus 13 or 47XX plus 13, a female or male with trisomy 13 or Patau syndrome. Then 47XY plus 18 or 47XX plus 18 trisomy 18 or the Edwards syndrome. So the human karyotype is abnormal if the chromosome is not 46. So abnormal chromosomes account at least 50% of spontaneous abortion. So euploid somatic human cells has 22 pairs of autosomes and 1 pair of sex chromosomes. So what are the chromosome abnormalities? Polyploidy, extra chromosome set, aneuploidy, monosomy or trisomy. Pag aneuploidy and extra or missing chromosome, mono, it means one chromosome absent, trisomy, one chromosome extra. Deletion, part of chromosome is missing. Duplication, nagdodoble. Translocation are two chromosomes join long arms or exchange parts. Inversion, segment of chromosome reversed. We also have isochromosome, a chromosome with identical arms. Ring chromosome, a chromosome that forms a ring due to deletions in telomeres. So chromosome abnormalities, we have deletion, naalis yung isang part. Duplication, nagdodoble. Inversions, nagbabaliktad. Substitution, and then translocation. So we have here AGIF. Next, GIF din yan of what's happening. For isochromosomes, yan yung mga ring chromosome. So comparison of trisomy 13, 18, and 21. Again, if we say trisomy, it means may one extra chromosome. So for 13, that's pataw. So incidence is 1 of 12,500 to 1 of 21,700. Then 18 naman is Edward. It's 1 of 6,000 to 1 to 10,000. Then 21 down. 1 to 802, 1 is to 826. So, mas mataas yung percentage ng mga incidents at birth ng Down syndrome. So, trisomy 21 or the Down syndrome is the most common autosomal aneuploid among live births. So, extra folds in the eyelids cause epicanthal folds, tapos flat face of a person. Tapos, naging term dun is mongoloid. Tapos, they resemble Mongolian in heritage. So, they are usually short, has straight hair and a tongue protruding through the thick lips. So, they also have floppy appearances. Then, developmental milestones like sitting, standing, and walking come slowly, and toilet training takes several years. So, intelligence varies greatly. Suppress din yung immune system nila. So, child with Down syndrome is 15 times more likely to develop leukemia. So, those are the pictures of children with Down syndrome. So, tingnan nyo to, oh, may extra na chromosome sa 21st chromosome. So, gene associated with trisomy 21, we have amyloid precursor proteins deposits in the brain. We have chromatin assembly factor 1 or CAF1A, impaired DNA synthesis, collagen type 6, heart defects, crystalline, cataracts, then crystalline beta synthase, CBS impaired metabolism and DNA repair, interferon receptor 1 impaired immunity. Also have kinase 1 for mental retardation, oncoprotein ETS2 skeletal abnormalities, phosphoribosyl glycinamide formyltransferase, impaired DNA synthesis and repair. We also have superoxide dismutase for premature aging. Let's proceed to trisomy 18 or Edward syndrome. So, affected children have great physical and mental disabilities. Tapos, they oddly have clenched fists, narrow and flat skull, tapos rocker bottom feet. Yun yung mga itsura. Ito, oh. Tapos, extra chromosome on the 18th. Then trisomy 13 or the pataw, most striking, although rare feature is the fusion of developing eyes. More common is small or absent ang eyes. Nose is malformed, may cleft palate or lip. Extra fingers and toes occur. Ganyan yung itsura niya. Turner syndrome, 45X. So, ibig sabihin ay absence of one X chromosome. So, Henry Turner described 7 young women aged 15 to 23 who were sexually underdeveloped. Then 8 years later, it was described by English physician Ulrich. So, it is called Ulrich syndrome in UK. So, meron silang slight webbing at the back of the neck. Tapos short stature. Sexual maturity, sparse body hair develops. But girls do not ovulate or menstruate and their breasts do not develop. So the uterus is very small, but the vagina and the cervix is normal. They are generally infertile. Intelligence is normal and the life can be fairly normal. Ang itsura nung may mga Turner syndrome. So extra X chromosome, also known as triple X, so tall stature and menstrual irregularities. So, about in every 1,000 males have an extra chromosome, which causes the Klinefelter syndrome. So, they may develop breast tissues, and most common genetic or chromosomal cause of infertility. XYY syndrome, 96% of XYY males are apparently normal. The only symptoms attributable to the extra chromosome may be great height and acne. Yan yun.